All right, hello everyone. Today we are going to be talking about protein synthesis. Let's begin with the basics. What is a protein? A protein is a polymer of an amino acid. Well, a polymer of amino acids. That means it's made up of multiple amino acids. What is protein synthesis? Simply put, protein synthesis is the synthesis of protein. Where does it occur? This is a multi-layered question, it's a two-layered question, but the synthesis of the protein itself occurs in the cytosol. How does it occur? It occurs in two stages, transcription and translation. Stage one is transcription. This occurs in the nucleus of the cell because, the nucleus contains DNA. What does DNA have to do with any of this? DNA has everything to do with all of this. DNA contains information. The information is coded in the bases of DNA. So, the base pairs of the DNA. Adenine is to thymine, cytosine to guanine. These sequences, these base pairs, are all sequenced in your DNA. So you just have long strands of this. You just have CCTA, long strands of this. So what happens is an enzyme called a helicase opens up your DNA double helix. This is your open double helix, the illustration on the left here. With this newly opened double helix, it's called a replication fork. An enzyme called RNA polymerase attaches and begins coding for the base pair of the now open DNA. So let's give you an example here. So your sequence goes as follows: adenine, guanine, cytosine, adenine. What would the RNA polymerase code for? Simply put, the opposite of this. The opposite of this sequence right here. Now, what would the opposite of this sequence be? You would think it's thymine, but no. In RNA, thymine is replaced with uracil. So it's uracil. What is the opposite of guanine? Cytosine to guanine, and then uracil. This is now your new mRNA. Now, three of these bases form what we call a codon. This is the information that gets translated into, translated into an amino acid that will then build your protein. So the RNA polymerase keeps on building and building these things called mRNA, messenger RNA. I caught the voice crack, don't worry about it. And, and they're called this because they carry information out of the nucleus into the cytosol. But then you're going to ask, why can't DNA just leave the cytosol, like into the cytosol from the nucleus? Simply put, DNA is far too big. It's double-stranded. It is far too big to just leave. So the mRNA strand, your mRNA strand, which is made up of these triplet bases called codons, then get checked by an enzyme. This enzyme removes the intron part of your strand. So what was our strand earlier again? Let's just make a new one. UUGCUGA. This is your mRNA strand, but not all of this is usable information. So let's just say this part is useless. That enzyme will then erase this part, the intron part of your mRNA sequence, before letting your mRNA leave into the cytosol. Now you're just left with the coding parts of your mRNA strand called exons. So let's just do a refresher here. Intron, non-coding. Exon is coding. I remember this with intron, intern. Interns don't code. That's how I remember it, at least. So now, stage one is complete. Your RNA has been made, your mRNA has been made, it's been checked, and now it's been allowed to leave into your cytosol. Once it leaves the nucleus, entering the cytosol, this is where stage two happens, translation. The traveling mRNA strand, let's draw your strand here, this, and then these are your bases, right? This traveling mRNA strand is surrounded by a ribosomal subunit. This ribosomal subunit will read this strand until it reaches a certain codon. It reaches a codon sequence of AUG. This is our start codon. What the start codon does, it basically tells the tRNA, the tRNA, to bring the matching anticodon sequence. You're gonna ask, what is an anticodon sequence? Well, remember, since a codon is made up of 3 bases, naturally these bases have a base pair. The anticodon simply carries the pair of that base that makes up the original codon. Why does it matter? Because a codon, here's the name, a codon codes for an amino acid. The tRNA carries the amino acids that we get from food and builds that amino acid chain longer and longer and longer, and this process continues until it reads for a stop codon. But by that point, you already have your protein. So what happens is, this is your start codon, this is your other genetic information. It will continue reading this like a typewriter or a scanner. This will keep going and going and going and going and going. For every 3, a tRNA flies in with your anticodon sequence that carries the amino acid respective to that anticodon. Okay? It carries the amino acid, and once it reads, it releases that amino acid and it builds and 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 builds until it reads your stop codon. It's a very, very like complex little process, right? But that is essentially what protein synthesis is. So let's do a quick recap. Stage 1, you have your DNA. Let me erase all of this. Let's do a recap. So you have stage 1. Stage 1. This occurs in your nucleus. You have your DNA, which will then be uncased, not uncased, but unraveled by an enzyme called helicase. Here, your mRNA will read the DNA, so you have your open DNA now. This is your open DNA. RNA polymerase will attach to this, and then it will start coding and coding and coding, making the opposite base pair until you get an mRNA strand. This is your mRNA strand. Once you have your mRNA strand, it will then be read again by enzymes that will remove the intron portions, the non-coding portions, the useless information, so that it can then leave into the cytosol, and then stage 2 begins. There, ribosomes will surround your exon-only mRNA until it reads for a start codon, which is AUG. Once it reads for AUG, this signals tRNA to bring the anticodon sequence that carries the amino acid specific to that sequence from your food, and then it will continue building and building and building and building and building, making a polymer, a chain of your amino acids until it reads a stop codon. Here, down here, I provided a table for your start, your stop codons, and which sequences code for which amino acid. But let's hope that you don't have to memorize all of this, because I sure as hell didn't. But that is essentially what protein synthesis is. If you have any questions, feel free to ask me. That's about it. See ya.